Introduce Léa El Khoury, and she's going to tell us about absolute binding free energy predictions for heat shock protein 90 complexes.
Thank you. Good afternoon, everyone. Today I will present the work we've done on heat shock protein 90 or HSP90 complexes. HSP90 is a molecular chaperone and one of the most abundant proteins expressed in cells. In normal cells, it helps in a proper folding of the protein and the degradation of damaged proteins. Under stress conditions, such as heavy metal or heat, it interacts with co-chaperones to form a superchaperone complex, which recognizes and stabilizes oncogenic proteins, such as protein kinases and transcription factors. Therefore, HSP90 has become one of the most important targets for new types of anti-cancer drugs. HSP90 consists on a highly conserved C-terminal domain, a middle domain, a highly conserved N-terminal domain. Our research focuses on the N-terminal part of HSP90 that binds ATP. Since the hydrolysis of ATP is a key step in the activation of the whole protein. Also, it has been shown that the inhibition of this site will directly lead to the downregulation of oncogenic client's protein. The ultimate goal of this study is to evaluate the binding affinities of small molecules towards the N-terminal part of HSP90. First, we want to test the accuracy of our predictions. That's why we chose HSP90 ligand complexes with known crystal structures and experimental binding affinities. These belong to three different chemical families: benzophenone-like, aminopyrimidine, and oxybenzimidazole. In the following, I will talk about one ligand, the work we've done on one ligand from each family, and I will name them A, B, and C. Since we want to compare the activities of ligands belonging to different chemical families, we chose to do absolute binding free energies. We are aware that relative binding free energies is less computationally intensive than absolute, but it would be so hard to transform these kind of scaffolds or ring to the others during the simulations. So we used Yank, a GPU-accelerated framework developed by the Chodera's lab. This automated package will allow the computing of absolute binding free energies in implicit and explicit solvent, as shown on this cycle. On the left, we have the protein and the ligand, fully solvated. On the right, we have the ligand bound to the protein, and the complex is fully solvated. So delta G is the free energy of forming this complex under these conditions. The unbound and the bound states are linked by a path that we choose, consisting of several alchemical steps that gradually eliminate the interaction of the ligand with its surroundings. In subsequent steps, this reaction is reversed in order to generate a fully interacting ligand with its environment and also completing the thermodynamic cycle. So on this figure, I'm showing the fully interacting ligand in green and the fully decoupled ligand in gray. Also during this cycle, we use restraints to keep the ligand in the binding pocket or near the binding pocket of the protein, and the free energy of restraining the ligand will depend on the type of the restraints. So we have two types of restraints: distance restraints and orientational restraints. Let's start with distance restraints, where we have a single distance between the ligand and receptor. This will allow the ligand to explore all the possible binding modes and all the binding sites without drifting away from the receptor. We have two variants of distance restraints: harmonic restraints, where the ligand can explore around the protein in a zone or a shell defined by a fixed radius; flat bottom restraints, where the ligand can explore anywhere in this spherical region. So the other type is orientational restraints, known as Borish style restraints, because these were suggested by Stefan Borish. We restrain one distance, two angle, and three dihedrals in order to keep the ligand in a specific binding pose inside the receptor. So we were curious to know if the type of the restraints will impact the values of the absolute free energies. Therefore, we used Yanks to test the 3 types of restraints on a host gas system. So what's a host gas system? Hosts are small molecules, usually fewer than 1 non-hydrogen bond atoms, having a cavity allowing them to bind small ligands called gases with a significant affinity. Why did we choose host gas system for our restraints test? Because hosts bind their gases with the same basic forces that protein use to bind their ligands. Also, their small size and sometimes their rigidity make it feasible to sample all relevant conformations. And the experiments can be done under conditions that make the protonation states of the host and the guest unambiguous. So we chose a cyclodextrin formed by glucose monomer as a host, bound to a cyclopentanol as a guest. And we did absolute free energy calculation using Yang with the 3 types of restraint: harmonic, flat bottom, and orientational restraints. We did 40 nanosecond per alchemical step or per lambda window in explicit solvent. And here I'm showing the free energies, and as we can see, with the different types of restraints, we are converging almost at the same time to the same free energy value. So the convergence of the free energy is independent in a host gas system of the type of the restraints. So after that, we wanted to choose 1 type of restraints to apply it on the main system we are studying, the heat shock protein 90 ligand complexes. We picked orientational restraints because we want to keep the ligand, so this will help the ligand not to sample all the relevant conformation and all the space in the simulation box. And from this study, we took the optimized alchemical step set that we tested with the host gas system to apply it on absolute binding free energies for HSP90. So during the simulations with compound A and C, we can see that the ligand is in the binding site with 1 binding mode for each of these ligands. However, for compound B, we found 2 different binding poses. It's true that we have restrained the orientation of the ligand relatively to the protein, but these 2 conformations are due to a rotatable bond inside the molecule. After that, we checked which of the 2 conformations we are mostly sampling during our simulations. So if we plot the dihedral corresponding to this rotatable bond, we can see that during the simulation we are alternating between the 2 conformations. So we are taking into consideration 2 binding modes while performing our absolute binding free energies. Then here I'm showing the values for HSP90 complex to compound A, B, and C with 20 nanosecond per lambda window in explicit solvent using Yang also, and with orientational restraints. As we can see, for compound C and A, we have a reasonable convergence for the free energy. However, for compound B, we have a slower or more difficult convergence, and this could be explained by the fact that this ligand need to be sampled, the simulation need to sample the 2 conformation of this ligand because it has 2 different binding modes. After that, we compared our computed free energies to the experimental values shown as dashed line on the graph, and the respective experimental values are here at the bottom of the slide. So we were able to compare our results with the experimental binding affinities, and we found the same ranking for the 3 ligands with an offset of 2 to 3 kilocal per mole. After that, we wanted to understand or to evaluate the factors governing these different efficiencies of these ligands. Since HSP90 ATP binding site is known to be one of the most difficult ones because we have protein conformational changes and charge side chains inside the binding site, we decided to start with a structural study to evaluate the changes and the interactions of the protein upon binding of ligands A, B, and C. So the HSP90 APO structure adopts an open conformation. The binding of ATP will induce this conformational transition to a closed conformation. And these changes happen in the sequence of residues 94 to 125, known as lid structure. Moreover, several docking studies showed that the preference of a ligand to bind to an open or a closed conformation depends on its chemical family. That's why we wanted to, since A, B, and C are from different chemical families, we wanted to see how they make changes in while binding to the protein. So the starting crystal structure of these complexes adopt a closed conformation, and this can be validated by these contact maps. So here I'm showing the residue-residue contact of the APO-HSP90, and here are the residue-residue contact of HSP90 complexed with compound A. These two contact maps are almost the same, with a different pattern at this level, corresponding to the residues 94 to 125, which corresponds to the list structure of HSP90. If we zoom in on this pattern, we can see that during the simulation, the lid structure of HSP90 has more contacts, residue-residue contacts, than the APO structure of HSP90. So this is the signature of a closed conformation of HSP90, and this lid structure is so flexible and active during our simulations. Also, compound B and C follow the same trend of residue-residue contact interactions as compound A. So the three ligands, A, B, and C, induced and maintain a closed conformation during the simulation. And since these belonging to three different families mediate the same conformational rearrangement as ATP, we wanted to evaluate their different efficiency by evaluating the interactions in the binding site at anatomic level. So if we compare the interactions of A and B with the residues in the binding site, we can see that both ligands interact with the same residues. With the purple bars, I'm showing the residues forming H bonds with each of the ligands A and B. But one important point is none of the ligands interacting with A and B belong to the lid structure that reflects this conformational change. So these ligands are able to maintain a closed conformation of the protein without even touching any of the residues of the lid structure. So if we evaluate the interactions of A and B in the binding site, let's take with the residues, let's take, for example, threonine 169. We can see that the distance between the carbon beta of the side chain of threonine 169 and each of the ligands A and B is almost the same. And this is the case of most of the residues interacting with A and B in a similar way in the binding site. However, we found some exceptions that may explain the better affinity of compound A. So we found two closer contacts of isoleucine 81 and glycine 82 with the aminopyrimidine moiety of compound A. And these are stabilized by two H bonds with the main chain and the side chain of isoleucine and glycine respectively. Also, we identified a sulfur-oxygen bond between compound A and the methionine 83, and a favorable positioning for a pi stacking between the aminopyrimidine moiety and the sulfur of the same methionine 83. Moreover, alanine 40, the most interacting residue with both the ligands, is closer to compound A than to compound B. So all of these extra contacts with compound A are due to its aminopyrimidine moiety that enlarges the contacting surface of this ligand inside the binding site. And this moiety is also present in ATP structure. So this makes compound A and the aminopyrimidine moiety a good mimic and inhibitor of ATP among the other ligands. One of the most important challenges during performing our absolute binding free energy is to take into consideration the different tautomers forms of the ligands we are studying. So in our case, we considered the neutral form since after analyzing the interactions of the ligand in the binding site in the first crystal structure or in the initial structure that we are studying. So, but we are interested in taking into consideration all the tautomers that may exist during the simulations. So, to conclude, we were able, by using Yang, to do absolute binding free energy calculation on host gas system and protein ligand systems. So for the host gas system, we found that the free energy convergence is independent of the type of restraints. For the protein ligand system, we found the same experimental ranking for HSP90 ligands, and the difference between our experimental values, and our computed binding free energy may be due to the parameters of the force field. We were able to sample during our simulation the two conformations of one of the compounds we are studying, and to explore the interactions of these compounds with the HSP90 ATP binding site. We believe that we will be able to see more relevant conformational changes in the HSP90 binding site in case we heat up a region, specifically the lid structure of this protein. As next steps, we want to compute the affinities of these three sets with the HSP90. These are from the D3R Grand Challenge, and most of them has a aminopyrimidine moiety as compound A. But we don't have the crystal structure of these compounds. Therefore, we want to follow this workflow by starting with a PDB reference using some docking techniques, then short MD simulations to eliminate the similar binding poses. After that, we will use some clustering technique like TICA and PCA used and tested in the lab to have a correct binding mode in order to compute absolute or relative binding free energies of HSP90 ligand complexes, also using Yang. So finally, I would like to thank the organizer of this workshop for giving me the opportunity to present out my work. I would like to thank Michelle Wren. She was a rotation student in the lab, and she worked also on the restraints project. I would like to thank John Chodera and Andrea Rizzi, NIH for funding this project, all the members of the Mobley's lab, especially my mentor David Mobley, and the NIH for funding this project. And I would like to thank you all for your attention.
Questions for Larry? Out in the back.
Yeah, thanks very much for the presentation. The relative ranking of the compounds was consistent right from the very beginning of your simulation, so even before you reach convergence. Is that common in other data sets you've looked at? Is that a feature that is often preserved? That the convergence, you get the convergence, but relative ranking was good right from the beginning. Yeah.
Yeah, the ranking was good from the beginning, but if we can see here, we have the compound C converging in another direction than these two compounds. So this was, I think it's not, or it's something curious to know why. So, but I like. So actually, it's if we compare the HSP90 to here also, okay, we have with the different restraints. Okay, here we go. Sorry. So we are asking me if it's common to see that we have the same ranking from the beginning of the convergence of our simulation.
Maybe this is one data set that
Yes, since it's the only data set that I have on HSP90, but I can talk about T4 lysozyme complex. So we are also studying T4 lysozyme, and we can see that the convergence at the beginning, not for the whole congeneric series that we are studying, is the same. But I cannot say because I'm just studying two types of molecules.
Christopher? I've never worked with HSP90, but if ligand binding causes lid closing, then I would expect that you might be able to see two-state binding kinetics to this enzyme, which should have separate on and off rates, so possibly separate thermodynamics. Have you thought of doing the free energy calculations separately to the lid open and lid closed forms, not allowing the lid to open or close, and then see if there is two-state binding kinetics, see if you can relate it to that?
Yeah, actually, it's a good point. We didn't think about it since we started from the crystal structure already in the closed conformation. But it would be interesting to see if these ligands will bind already to the open structure by using. We cannot do it if we don't have a good docking approach to see the comparison of if it because I saw several docking studies where they are showing that some ligands can bind to the two structures, the apo and the open conformation and the closed conformation. But some others don't have this possibility, like the ligands we are studying. So these in the beginning don't have the possibility to bind to the unopen conformation. So that's why it would be a possibility if we heat up this structure to see if we will have this transition and if these ligands will stay stable in an open conformation, or even if we will see this open conformation during the simulation. So yeah, it's to see.
A few things. First of all, the actual structure of HSP90 in solution in APO form, it's still very much an open question. We can discuss a little bit more about that later. But okay, the crystal structure in APO form is the one you study, but when you perform biophysics, it's pretty complicated. Then you might be interested in a pretty big data set that relates to HSP90 that has been released, that is being released due to the K4DD EMI consortium, where there have been plenty of structure, thermodynamics data and kinetics data generated on this very system. So. Okay.
Yeah, and what I would be interested to talk about the different structure of APO, because the main reason of a closed conformation was the binding of the ATP. And here we are studying just the APO structure of the N-terminal domain, not for the whole, and on a monomer. And this will vary if we are studying the dimer. We will have a lot of conformational changes between APO and...
yes, I'm talking about that.
Yes, okay.
Any other questions? Let's thank Leah again.